Hi everyone, I hope that you're doing well. So today we are back with another topic that is GPCR. GPCR stands for G-protein coupled receptors. Fine. So GPCR stands for G-protein coupled receptors. Let's talk about what is this G-protein. So these are guanine nucleotide binding proteins. Fine. So in short form, it is known as G protein. Okay. So we got to know about G protein. Coupled receptors means this is associated with the G protein. Okay. Guanine nucleotide binding protein. Okay. Clear? Okay. So what are receptors? Receptors are those molecules which are present on the surface of a cell. It accepts the signal or it recepts the signal. It is meant for the reception of signal. That signal, that whatever signal would come would be recepted or it would be accepted by those receptor. And once this signal has been accepted by those receptor, that would go inside the cell. Fine. So what could be your signal? Signal could be your hormone. Signal could be a drug. Signal could be a neurotransmitter, or it could be a amine. It could be a lipid, etc. Lipids, etc. Okay, so we would be having a lot type of signals. That could be hormone, that could be drug, that could be neurotransmitter, that could be amine, that could be lipid. Fine. I'm so sorry. So what happens if we take an example of hormone? So we have hormone known as epinephrine. So the reception of epinephrine is through our GPCR, and we have a neurotransmitter known as EGF, that is epidermal growth factor. So these are recepted by GPCR. So let's talk about the structure of GPCR, that how exactly our GPCR looks like. So let me just zoom it out so that it would be clearly visible. Fine. So this is our GPCR. So here our ligand is our epinephrine. But let's not talk about ligand here. We're just talking about the GPCR, our G protein coupled receptor in resting state. So as you could see, this GPCR is a 7 transmembrane structure. Okay, we have 7 transmembrane. We have extracellular transmembrane, extracellular membrane, we have intracellular membrane. So this in this extracellular membrane we would be having nitrogen free terminal. So the terminal would be of nitrogen, and here the chain would end with the free carbon terminal. So we would be having free carbon terminal here. And this 7 transmembrane structure is also called as beta endogenic receptor. For the epinephrine, it is beta endogenic receptor, and its name changes with the hormone or ligand we have. Okay, so we could call it as beta receptor if it is like it is adrenergic, adren, adrenergic. Okay, so I'm so sorry, androgenic, that is different. Okay, so what happens if it is from the adrenal gland? It is adrenergic. Okay, if it is from the, for example, it is a neurotransmitter from the epidermal growth, pituitary or something. Okay, the name would be according to that. Okay, so we have extracellular surface, we have intracellular surface. Now we have a G protein associated in the intracellular space, that is associated with the cell membrane, as we could see here. Now this G protein, this is the main thing here. It is a trimeric protein. Trimeric means it contains 3 subunits. What are those 3 subunits? This subunit is known as G alpha. This subunit is known as G gamma, or this bigger subunit is known as G beta. So in resting state, these are trimeric like this. This is your alpha, G gamma, and G beta. Fine. And you could see we have a adenyl cyclase, which is a secondary messenger. Fine. So what exactly we talked about? We talked about the general structure of GPCR, that is G protein coupled receptor. Let's talk about GPCR in bit detail. So G protein coupled receptor, GPCR is also called as 7 transmembrane receptor or heptath helical structure. Protein located in the cell membrane that binds the extracellular substances and transmits signal from these substances to an intracellular molecule, that is a G protein. So as we have seen here, the signal would come from here, it would be transferred to the G protein. Fine? Okay. Now, if you don't get anything, just post a comment. I'll explain in the comment section, or I'll make a further video that would be a doubt clearing video on GPCR. Fine. So it happened till here. We talked about that we have an intracellular signal or intracellular transmission of signal through G protein. So G protein are associated with the intracellular transmission of G protein transmitted signal. So signal would be transmitted from G protein to our secondary messenger, that is adenyl cyclase. Fine. So that would be our secondary messenger here. Now we have GPCR in variety of organisms, including mammals, plants, microorganisms, invertebrates. Okay. Even we have a wide variety of GPCRs. In humans, there are more than 100 types of GPCR or 100 types of genes, or we have 100 types of gene encoded GPCR. Okay. As we know that everything is gene encoded. So gene encodes for a particular thing. Our receptors are protein also. Okay. So what are those ligands? What could be your ligands? As I've told you, this ligand could be your hormone, it could be your drug, neurotransmitter, amine, lipids, etc. And it could be your light in case of rhodopsin in rod cells. Okay. The receptors of rod cell would be having GPCR. Now what happens? We know that the rod cell has rhodopsin. Once the light would fall on the proteins, the protein present in the cell would be rhodopsin and the receptor would be GPCR. Once light falls on it, it start activating, and what would be the effect? Yes, we would be having vision. Okay, so effect here would be vision. When we are taking the case of epinephrine, epinephrine is a fight and flight hormones. What would happen? Once your epinephrine would bind to this receptor, that would activate the secondary messenger. The secondary messenger here is your CAMP, cyclic adenyl monophosphate. Okay, CAMP pathway would take place, and that would, what that would do? That would increase the heart rate. Fine. Okay. So we talked about the ligands here. Ligand could be hormone, light, amine, neurotransmitter, lipids. It could be anything. Few examples: we have prostaglandin, we have epinephrine, we have rhodopsin. Okay. So who discovered this GPCR? So this GPCR is discovered in 1970 by American physician and molecular biologist Robert L. Lefkowitz. Lefkowitz shared a Nobel Prize in 2012 with a chemist known as Brian K. Kobelka. Okay, so they actually discovered the structure and function of GPCR. They got a Nobel Prize in structure and function of GPCR, right? So we talked about the resting stage of GPCR. Let me erase it so that we could be having a stimulated phase of GPCR. Okay, fine. I'm just erasing everything so that we could be having a stimulated phase. For example, our epinephrine is secreted from the medullary region of adrenal gland. Okay, adrenal gland secreted epinephrine. We have two parts of adrenal gland. We have medulla, we have cortex. So cortex secretes glucocorticoids and mineralocorticoids, while your medulla secretes epinephrine and norepinephrine. Fine. So in resting state, we know that this G protein is a trimeric structure which is associated with the adenyl cyclase. Fine. So what happens? This epinephrine has been secreted to the Yes, it is secreted in the bloodstream. Now it reaches to the effective organ, for example, the heart. Okay, or we could say the pacemaker, say no atrial node. It would means it would reach to the sinoatrial node. Now the surface of the sinoatrial node has this membrane receptor known as GPCR. Now this GPCR binds to transmembrane structure. Okay, beta adrenergic receptor. So it binds to the beta adrenergic receptor. Now what would happen? As this epinephrine binds to the surface of the receptor, it would all of a sudden what it would do? It would activate GEF, that is guanine nucleotide exchange factor. Fine. It would activate this guanine nucleotide exchange factor. What it would do? It would convert GDP into GTP. Yes, into GTP. Oh, okay, okay. I'm so sorry. I'm so sorry. Okay. Actually, this is vice versa. It is opposite of this. This GTP would be converted into GTP. GDP, right? What would happen here? One more thing is that this G protein, when it is in a resting state, there would be GTP present. Okay. Now what would happen? This dephosphorylation would release one phosphate here. Okay. We would be having one phosphate here. Okay. Fine. So what happens? This GTP is converted into GDP. Now this GDP, what would happen? This GDP would cleave this trimeric protein. In trimeric protein, we had G alpha, G beta, and G gamma. So what would happen? This would cleave this G protein from here. Now we would be having V alpha and GB. So I'm not B, it's G alpha, G gamma, and RGDP. So they would be joined together, and as the cleavage take place, this G, G beta would be attached with our adenyl cyclase. Now, this adenyl cyclase, what it would do? Once G beta has joined to the adenyl cyclase, this adenyl cyclase, this would do further dephosphorylation, of ATP. It would change it into AMP, cyclic AMP. This, what would happen? How adenyl cyclase would activate? It would activate by G beta and adenyl cyclase. Okay, it would cleave this ATP into cyclic AMP, and it would act as a secondary messenger to the cell, and it would result in the increased heart rate. It would result in the dilation of skeletal muscle. So this dilation of skeletal muscle would help, means this would occur by extra phosphates. We have phosphate released in this step and phosphate released from this step. It would do that too. And it also breaks glycogen into glucose so that we could run, we could fight. Fine. So this glycogen into glucose also done with the help of phosphate that has been released. So we got to know about GPCR. Okay. Now we have a further information about GPCR, and we'll revise this GPCR. GPCR is made of three basic protein that has three basic regions: extracellular portion that is of the N terminus, free N terminal; intracellular portion would be having free C terminus, and middle segment containing 7 transmembrane domains. Beginning in the N terminus, this is the long protein winds up through the cell membrane. Okay, we know this right now. Now we know it, right? So I don't think if you wanna start if you wanna read to it, you could just pause the video and you could read it, right? So we could see that epinephrine change in the vision of means we have a ligand, right? So ligand is required. Ligand could be your sunlight, ligand could be your epinephrine, ligand could be your drug. See, basically this happens. I just explained you in the case of epinephrine. Okay, if like this would be your rod cells, or it would be a rhodopsin protein. What would be happen? Rod cell would elongate. The elongation of cells of rod cells of eye would be due to the activation of secondary messenger, that is our CAM pathway. Okay, fine, very good. Now we have both inborn and acquired mutation in gene encoding GPCR can give rise to disease in human. Okay, so it also associated means the malfunctioning of GPCR. Malfunctioning of G-protein coupled receptors could result into disease in human. For example, if the mutation of rhodopsin result in the continuous activation of intercellular signaling molecule, which cause the congenital night blindness. For example, if your genes encoding for GPCR are not working, so what would happen? Sunlight would fall on the cells, right? Or light would fall on the eye cells, but it could not activate our secondary messenger. Hence, we could not see in the light, or we would be having night blindness. For example, in addition, acquired mutation in certain GPCR cause abnormal increase in receptor activity and expression in cell membrane, which can give rise to cancer. For example, what happens if your GPCR not working properly? So we would be having a lot continuous of the signals, right? Cause abnormal increase in the receptor activity. It would result into cell division. As we are getting continuously, we are getting signal, continuously we are getting signal, so protein synthesis would take place, as it would direct cell that make proteins and undergo cell division. Okay, in case of schizophrenia, like we have two neurotransmitters are dopamine and serotonin. By blocking the receptor drug, what exactly happens here, like this antipsychotic drug, your antipsychotic drug which is used, this is colzapine and olanzapine, which blocks the GPCR that normally binds the dopamine or serotonin. What would happen? These antipsychotic agent would bind to a GPCR, and it would result in the it would depress our schizophrenia. Okay, or it would block the signals which are continuously coming from our ligands, right? What ligands? Dopamine and serotonin, which is doing or which is causing that mental disease known as schizophrenia. Fine. Even drugs salmeterol and albuterol, we know that we used them in COPD and asthma. So what exactly they do? They block this GPCR. Once this GPCR-G-protein coupled receptor has been blocked, now what would happen? This blocking would result in the depression, or it would suppress the inflammation of our lungs. Okay, during what happens, that inflammation caused our bronchi are inflamed. So what would happen? It would result in the blockage of inflammation of our lungs. Fine. So these drugs, salmeterol, as I told that your ligand could be salmeterol, albuterol, even it could be your clozapine or olanzapine, or it could be your other other mutations. Okay, or it could be your other ligand, right? Ligand could be anything. It could be your hormone like epinephrine. It could be your neurotransmitter like dopamine and serotonin. It could be your drug are this clozapine, olanzapine, and your salmeterol or albuterol. So this action of drugs, a few drugs like salmeterol, albuterol, clozapine, and olanzapine is by blocking this GPCR, G protein coupled receptors. So that was all about GPCR, G protein coupled receptors. In the next video, we're gonna talk about CAM pathway, that is cyclic adenosine monophosphate pathway, that how it acts as a secondary messenger. Till then, take care of yourself. Have a great day ahead. So we'll meet in the next session. Okay, if you have any doubt, just comment it down. I'll explain it to you on the comment section. Okay, even if you don't want to comment, then what? Then you can write to me. Okay, just write to me. I'll make another video where I could clear your doubt. Fine. So I'll leave my mail ID in the comment section as well, or in the description. There would be my email ID. Just give me a mail, and there we could talk about your doubt. Fine. So by this, I'll end up this discussion. I hope that you have liked it. If you have liked it, if you have understood the concept, so please hit a like and please subscribe to the channel. Fine. And don't forget to press the bell icon so that next video, the CAM pathway video, would not be missed. Fine. Bye bye. Take care. Have a nice day.